Okay, this one's been requested about gas exchange, and it's a large topic. So it was really looking at insects, so about a tracheal system in insects, so trache, tracheoles, and spiracles. We need to know what those three words mean. And fish, the fish have got gills, they have gill lamellae and filaments, and the countercurrent principle. Um, and then it says a structural and functional compromise between the opposing needs for gas exchange and limitation of water loss shown by terrestrial insects. Obviously, it doesn't say fish. Fish aren't bothered about water loss. I'm sure we all know why. So let's take insects first. So I've got a little diagram. Again, I've gone for the simplest as possible. So you can see here that here's outside of the insect, and so this is the air outside, and they have a tracheal system. So they have a spiracle, which is a bit like a stomata, in that it can open and close. Then we have the trachea, which is just like our trachea. It's got circles around it. It's like those lines are down it to show that it's reinforced. Because when air moves through tubes, it can suck the walls in, and therefore it needs reinforcement. And they branch and branch and branch and branch, and they then end up in these microscopic little tubes called tracheoles, and then they terminate where the cells are. So if I can pop some cells on, do a little bit of a drawing. So then this is, I don't like green. We'll have a pick green there. Let's go for black. Black, always best. So there's some cells. So the body cells. So you can imagine that all our cells are surrounded by fluid, and it's no different in insects. And if we zoomed into these tubes, you can see that the tracheoles. Let's leave the label on so you don't forget. I mean, it is poor that they've labelled this diagram with arrowheads. Remember, when we label in biology, it should be a straight line touching the thing that we are labeling, no arrowhead on it, and then we label it. So it should be like that, but what can you do? Anyway, yeah, water or fluid. Yeah, so all of these cells are surrounded in fluid. Yeah, it's a fluid environment. So all of this is fluid. It's fluid, 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 fluid. So inevitably, it leaks back into these tracheoles. So all this is tissue fluid, it's called. The fluid that surrounds our tissues. Again, no different in insects. So there's a little bit of fluid in there. And for gases to get to these cells, well, there's fast diffusion through the air in the tracheoles, but then it has to dissolve to get from here into the water and then from there into the cell. And then, so that'll be oxygen moving that way and then CO2 moving back. Okay, so that's a brief overview. So you need to be able to describe spiracle. It's the opening of the trachea into the insect, and it can open and close. The trachea is a tube that allows air to move into the body of the insect, and then the tracheoles are branches of those that increase the surface area, so that the clusters of cells all have a delivery system for oxygen and a removal system for CO2. Right, anything to do with gas exchange relate to Fick's law, okay? Be it mammals, um, or fish or insects, and this is just that the rate of diffusion is proportional to the concentration gradient. So we need to think, how do insects maintain a concentration gradient? And times by the surface area. How do they ensure a large? Because I want this number to be big on top. I want a big concentration gradient. I want a big surface area, and I want a small thickness of barrier, so that I get a big number times a big number, gives me a big number, divided by a small number, therefore I get a bigger number possible. Yeah, if I want a low rate of diffusion, I need a zero gradient, a very small surface area, and a very large barrier. Okay, but for gas exchange in insects, they want it to be fast. So how do they get a fast concentration gradient? Sorry, a steep concentration gradient. Well, they can kind of pump their body, and they basically squeeze this. So they when they pump their body, they are squeezing these tubes, which acts like a bellow. So it helps to move air in and out. So that's concentration. Diffusion distance. Well, insects are small to start with, as we all know. Some spiders are worryingly big, but on the greater scheme of things, they are small, and they're limited by how big they can get. But this bit here, this is crucial for the diffusion distance. That diffusion through air is fast, diffusion through liquid is slower, and therefore this distance here, how much fluid is in the ends of the tracheoles is critical. And questions can sometimes ask, what happens if there was more fluid in this tracheole? Maybe in a certain condition it kind of led to it being that much. Well, it would take longer for the oxygen to diffuse through that bigger distance. Okay, so yeah, the thickness of the barrier really depends on how much fluid there is in the ends of these. And surface area, there's each cell or each cluster of cells that has its own tracheole. So because of the tracheoles branch, there's a large internal surface area. So just zooming back to this equation quickly, concentration gradient, well, for insects, they do this abdominal pumping, not on the syllabus, might appear in a question. Surface area, it's the structure. They have lots and lots of tracheoles, okay? Therefore, a large internal surface area. And thickness of barrier, the insects are small and they've only got a small amount of fluid in the ends of the tracheoles. Okay, so let's look at a graph, because in the exam they tend to ask about graphs, or tables of data. So here I've got time on the bottom, so I'm going to put an insect, monitor insects over time, and then I've got concentration in tracheoles, low and high. Red's going to be oxygen. So let's put a line on for oxygen. So, and the yellow bits are when the spiracles are open, and the white bits are when spiracles are closed. So when spiracles are closed, it can't get oxygen. So let's just start an arbitrary value here. Let's say it's got that much oxygen inside its tracheoles. If its spiracles are closed, well, it can't get any more oxygen in, and its cells will be using oxygen. Therefore, there'll be a decrease during that time. Then when the spiracles open, diffusion can take place, and therefore oxygen can come in, and therefore it will do, comes back in again, and then it goes down and goes back up again. So it's the equivalent of me holding my breath and then breathing and then holding my breath again. So when its spiracles are closed, it can't get any oxygen. So it's like, what's the point of having spiracles then? Why not just have its spiracles open all the time? Well, that's because of water vapor inside the tracheoles. Remember, insects, living things in general, are worried about water. Again, fish aren't, but if you're above ground, then you're going to be worried about that. So let's again just put an arbitrary. Let's start in the middle here, just to keep away from that red line a bit. When the spiracles are closed, well, the water vapor in the tracheoles is going to increase because it's going to evaporate from the cells around it. But when the spiracles are open, it's going to, and therefore going to lose that water. Then when the spiracles closed, water's going to go back in there, and so on and so on. If the spiracles were open all the time, it would just continually lose water. So by closing its spiracles, it means it can conserve water. So going back to the diagram for one last time for insects, because this is a speed through. So there's our spiracle. It's how it minimizes water loss. Okay, so it opens its spiracles to allow oxygen to diffuse in. Well, air will move in, but yeah, it's the diffusion of oxygen because oxygen is being used here. Therefore, there's a concentration difference. So oxygen diffuses from the air to the cells, and CO2 will diffuse from the cells back out again. If this insect lives somewhere like a tropical forest where it's 100% humid, in which case they can leave the spiracles open all the time because it's never going to lose water. If it's 100% humid outside and 100% humid inside, then it won't lose water, and therefore they can get oxygen all the time. And therefore insects in tropical forests can get bigger. If you want to be scared by a massive insect, go to a tropical forest. Yeah, it's nice and warm. They don't struggle for water. Therefore, they can leave the spiracles open. They can therefore get oxygen continuously. Therefore, they can do aerobic respiration continuously. Therefore, they can grow bigger. And therefore, you get these enormous beetles that live in forests. Not any old forest. It has to be these warm, tropical forests. Okay, so that's insects. Under 10 minutes. I've done well. After a quick sip of coffee, I'm back for gills and fish. So we need to know what gill lamellae are, filaments are, and what a gill is, and the countercurrent principle. And again, relating it to Fick's law. So again, I've drawn this picture of a gill. So this is a gill. This is 1 times gill. Okay, fish tend to have pairs of them, and they'll have like 4 pairs of gills. Okay, and this is a gill filament. So that's a filament, and on the filament are the lamellae. So I'm pointing to 2 here. So these are the lamellae. Pen's not working quite well. So those are the lamellae. So again, how do fish have a high surface area? Well, they have many gills. Each gill has got many filaments, and each filament has got many lamellae. Okay, therefore it's got a large surface area. And if we look at one lamella, kind of in 3D, these kind of look like like that. So there'd be almost like a plane wing coming out. So that's the filament. Then on the filament, there'd be all of these lamellae that basically stick out, and water's going to be moving across these lamellae. So in terms of thickness of barrier, well, the water is right next to the lamellae, and in the lamellae are the capillaries. So this is a capillary if you're American, and this is that gill lamella. So there's many lamellae, so this is one of them. Okay, and you can see that the blood's going from A to B, and crucially, water moves in the opposite direction. So water will move this way. We don't need to know about how fish move water. Again, if you're doing all past papers, there'll be questions about the opercular cavity and things like that. But we don't need to know that. They've stripped it out, where we just need to know about this principle. So if I take A and B, so this distance across the lamellae from A to B, and I put it onto a graph. So distance along gill lamella. This is oxygen concentration, and we're going to put 2 lines on. So let's go for blood first. So let's do for oxygen in blood. So I'm going to go for purple for blood. So at A, well, this is the blood arriving at the gills. It's therefore going to have a very low oxygen concentration because it's been round the fish's body. Okay, and as it goes across the gills, the oxygen in the actual blood is going to increase because it's going to move from the water into the blood. So it goes up. Okay, and then let's do water. Do that in blue because obviously water is blue. Well, at A, again, and this is where it's tricky, in that. Oh, I need to put my arrow on here as well. So yeah, so the blood's moving that way. And there's our water line. Let's put it on first, and then we can worry about it. So yeah, this is why, and it's easy if you think of at B, water's going to have 100% oxygen in it. Okay, it's just arriving at the gills, and therefore, it moves down a concentration gradient, and therefore it will move into the blood. As I move down to the center point about here, then the water has lost some oxygen, but it's still more than the blood, and therefore there's a diffusion gradient. And same at A, even though by A the water has lost a lot of oxygen into the blood, because it's reaching the blood at A that's got no oxygen in it, practically, there's still a diffusion gradient. So this is the counter-current flow principle, or counter-current flow mechanism. So counter-current with an E, flow. And it just means, yeah, so I would say in the exam that blood and water move in opposite directions. I have to write to think. Therefore, there's a diffusion gradient for oxygen along the entire length of the capillaries, or capillary in the lamella. Crucially, I need to say along the entire length. Okay, so opposite directions of blood and water, and yeah, along the entire length of the capillary. I also need to mention that as well. If they went the same way, so there's A and B, and in comes my so here's A, and I'm starting with the blood coming in, and therefore, well, it would increase and it would reach a 50% value. So that's kind of 50% in terms of oxygen concentration. And if I put the water on, well, water's going to come in at 100, and it's going to diffuse in, but it's going to reach equilibrium. They're going to get to the same point where there's just the blood's next to the water, and they've got the same concentration. Therefore, you'd only get to 50% concentration. Whereas here, in this example here, I'm getting to a higher, I'm getting to like 95% concentration of oxygen inside. So we'll just zoom back quickly, just to check that I've done everything. Oops, struggling to pinch and zoom. Let's pop it there. So yeah, I mentioned gill lamellae, filaments, countercurrent principle, or I put countercurrent flow mechanism, it's the same thing. And crucially, for fish, concentration gradient, well, we don't learn about ventilation in fish, so you can just, it's the countercurrent flow mechanism. That's how they maintain the concentration gradient along the entire length of the gill lamellae. Yeah, the capillaries in the gill lamellae have a constant concentration gradient due to the countercurrent flow mechanism. They have a large surface area because they have many gills that have got many filaments that have got many lamellae. Okay, and thickness of barrier, the lamellae have capillaries on their surface, and that surface is right next to the water. So there's a very small diffusion difference. It's just the same as we get in our alveoli. In our alveoli, we've got those cells that are kind of like, flattened cells, like that. Need another set here. So here's the alveoli. So this is the air, and this is the capillary. Therefore, there's a short diffusion distance, and it's just the same for the fish, except they don't even they are missing kind of that layer. This is just water. Where's my blue? This just becomes water. Finish with a messy scribble, but it's a good way to finish.